I'm going to talk a little bit about our work looking at translocation carriers. And we've been doing this for 5-6 years, Genesis Genetics. I myself am a cytogeneticist, so this is my pet subject, so I'm going to hopefully give you some information, but not drown you in cytogenetics. So this is my disclosure slide. I'm the laboratory director of 2 Genesis Genetics labs in the UK, also involved in the commercial side of Genesis Genetics in the US, and I'm a scientific advisor to a biotech called Rubicon Genomics. And in case anybody's had their head under a stone for a little while, Genesis Genetics is now part of Cooper Surgical, together with Recombine and Reprogenetics. So, reciprocal translocations, these are pretty prevalent in the normal population, although nobody really knows how prevalent in the normal population. Numbers vary when you look at the papers, between 1 in 500 and 1 in 600 of the normal population. These people will come along to the clinics. Sometimes they'll know they're carriers, sometimes they're not, and they have no phenotypic effects for the majority of the carriers of these translocations. The real problem is the increased risk of miscarriage. If you don't inherit the right proportions of those chromosomes with those translocations, then you have a very large chance of miscarriage. However, depending on families, they can be inherited, and you can pass them down successfully through generations. And there was a very nice study that looked at family trees and traced a translocation that formed back in 1752 in Finland. So they're out there. What really is key is where these translocations, where these chromosomes break across the different chromosomes. And there can be risks that vary from risk of miscarriage to, at the most severe form, risk of affected children. And that's something that is the real key issue with some of these translocations. They can also spontaneously occur in the population. So somebody can, they can pop up, and they tend to occur. And with those ones, you can also, depending where the translocation is, you could translocate breakpoint across a gene, and that gene function might be impaired, and that can lead to developmental delay or mental instability or all sorts of issues. These de novo cases we don't really see as PGD cases. So just to kind of give you the brief what happens, so we have our 2 sets of parental chromosomes. This is on top, the yellow and blue are some normal chromosomes, and at the bottom this is a carrier. So what part of the red chromosome is transferred to the green chromosome and vice versa. These patients have a normal full complement of their DNA. It's just the position of that DNA is swapped between 2 chromosomes. That doesn't affect them. They are phenotypically normal. However, unless you inherit the 2 normal copies of your chromosomes, a red and a green, or the 2 abnormals together, you end up with an embryo that has too much DNA or too little DNA. Once you get too much or too little, that's when you get the chance of miscarriage. So that's our real issue they're trying to look at. This is from an organisation called Unique in the UK, and they have very nice patient literature for this stuff and very nice advice leaflets if you need anything like that. Robertsonian translocation carriers, these are slightly different. So our chromosomes, you have the chromosomes that you kind of typical imagine picture of a chromosome with a little P arm and a Q arm. And some of the chromosomes are what we call acocentric. These are like 21, which has no P arm. And these kind of small little residual P arms can fuse together to produce a chromosome that has 2 long arms together. And these are Robertsonian translocations. They're a little bit rarer in the population. They are chromosomes 13, 14, 15, 21, 22. Generally, again, these people will come to your clinic. They have no presentation, apart from when you look at their cytogenetics. Again, there's an increased risk of miscarriage, but obviously as well, if you're looking at 13 and 21, there's an increased risk of Down syndrome or Pater syndrome. And the only other risk you have, if you inherit the same chromosome and you have a normal number, but you inherit 2 copies from your mother or father, then you can have what's known as uniparental disomy, and that is a negative effect as well. So when I looked at this literature, when Simon twisted my arm, don't know where he is, to give a talk, I looked at the literature fairly recently, and actually the literature still has lots of people still using fish to do translocations. And there's publications out there from 2016. This was, a technique that has its advantages, but like many things, not going on to fish and PGS conversations, we've moved on. So since 2011, we've been using array CGH to look for translocations. We use an array from a company called Bluenome that's out in the past that I used to work for, and it was a higher resolution array CGH that could look for these imbalances. There's some very nice papers back in 2011, 2012, 2013 that used this and some other techniques that looked at all the chromosomes. And this was a kind of nice advance from fish, which I'll come on to in a moment. So this was, I don't think Francesco Fiorentino was here, but this is a collaboration I did with Francesco Fiorentino back in 2011, which we published. And this is one of their cases, and this is an array CGH profile. So we have all the chromosomes, 1, 2, 3, up to 22, X and Y. And this was a 318 translocation that we looked at. And you can see that it has no changes in all the chromosomes apart from 2 small changes. One very small change on the top of chromosome 3 and one on chromosome 18. As always, this was the publication. This was the nicest picture we ever got. And this was the smallest imbalance that we found. And this was a trophectoderm biopsy. And we looked at it, and it was about 2.5 megabases. And it's the one that you always show to try and impress people. So it's a nice picture. But you can see that we're able to really identify these imbalances very accurately. And when we looked at this, there's a large table. So just to kind of give you the skinny on it without going through the whole table, I've just highlighted it. When we looked at the Robertsonian translocations on the left-hand column, just under 2/3 of those had aneuploidies. There was an average age of 38, so a little bit less aneuploidy than you find in the normal IVF population, but still finding aneuploidy in translocation carriers was still an interesting finding back then in 2011. And the reciprocal translocation carriers, they're about the same age and had a little bit more aneuploidy, although this was very small numbers back then in 2011. And when you look at the number that are suitable for transfer, from the Robertsonians, about a quarter of the embryos, 27% were suitable for transfer, and in the reciprocals, about 10% were suitable for transfer, which is an important finding because, if your patient's coming in, they're going to need 10 embryos before they find one suitable for transfer for reciprocal translocations. And this was a really small number, but the thing that was really great from this study is it had a great pregnancy rate, clinical pregnancy rate, and that was a big advantage. The real thing that you're trying to avoid with the translocation carriers is miscarriage. And these people go through sometimes horrendous cycles of miscarriage after miscarriage. And in this study, they'd only done 12, but they didn't have a single miscarriage. And that's one of the real advantages of this technology. So at that point, the real finding was that aneuploidy was still significant in translocation carriers, not just the general population, and that between 15 and 25% of the embryos would be suitable for transfer. So we did some work ourselves. The numbers on the left were some numbers from Genesis in collaboration with Care Fertility in Nottingham, and we found approximately the same number of embryos suitable for transfer. That's the top red number. And then about a little bit more aneuploidy, and our clinical pregnancy rate was a little lower, but these were very preliminary numbers. but it gave us enough information to go on to offer this as a service and to use it. So why change from something that was obviously working, that seemed to solve the problem of clinical pregnancy rates? Well, this new technology came along a couple of years ago, next generation sequencing, and it potentially gave us the chance to produce an even better test in accuracy, sensitivity, and specificity. Also, I showed you the nicest picture. Sometimes these were not very nice, and even with array CGH, it wasn't the latest technology that we could use. So we looked to try and move on and actually get an indication of better quality. The other thing that we wanted to do was to have a single workflow in the lab, so that if somebody came in for just general IVF, PGS, or they came in as a translocation carrier, we could use the same workflow. And in 2014, we did a validation experiment. So the translocations, being my pet subject, I find them really, really very interesting, because it's really pretty much the only instance in this kind of pre-implantation genetics field that we know what we expect to find before we start. So you get a carrier type from a clinical lab, and you know that in about half the embryos, if it's a reciprocal translocation, you're going to find an imbalance at 2 specific points. And that's really nice for validation experiments, and you always got a kind of internal validation for every experiment you do. So that's a real useful tool. So it was good to look at the sequencing, but it was also good to look at this as a service. And we did a comparison of the NGS with the array CGH. Of course, as well, we have the joy of being CAP, New York State, ISO 15189 accredited. So you need to do this in order to bring in a new service. So as always, you show the nice pictures. So this was a normal. This was a very early sort of data. This was an array CGH at the top from the same whole genome amplification. This was the early sequencing data. The difference being at the bottom, sequencing is complete copies. So in the midline across all 1 to 22, that's 2 copies. You have 1 copy of X, 1 copy of Y in this, so we know it's male. So what do we do? Well, we did a nice study using what is now our standard NGS platform, which I'll not really go into for time reasons. We took 191 embryos from structural abnormality carriers. That was 26 reciprocal translocation carrier families, 2 Robertsonian translocations, and 2 inversions. So these had been run by array CGH. We blinded it, and we ran them again and scored them by NGS. Even with reciprocal carriers, translocation carriers, you get embryos that have huge numbers of aneuploidies, and this was an instance here. This was the array at the top, and this was the sequencing at the bottom, and you get the same pattern forming. This is when it starts to get more interesting, of course. This is when we're looking at the translocations. So in this particular instance, at the bottom, this is a 3-5 translocation. You might be able to see here that this is a breakpoint on chromosome 3, and there's a smaller breakpoint on chromosome 5. It looks like a little sort of step change there. That was an early version of the software that didn't really work as smoothly as it does before. And you could see on the top one here that you have the same thing. The break on chromosome 5 is possibly a little bit more difficult to detect using the array CGH. And then you start to get other samples. So this was a 3-9 translocation. So we have 2 imbalances here. And you can see that this is actually much more indicative of what these early arrays that we've been using for many years are like. A little bit noisy, a few bumps, and a little bit more difficult to detect. But essentially they still work, but with some more noise in the system. So all the results obtained from the NGS were concordant with the array CGH, except in 2 cases where we had some small imbalances that weren't picked up. So you expect to see 2 imbalances in the translocation carriers. One of those imbalances was very small in a sample, which we missed by the array CGH, which we saw by the NGS. And that was a real clear indication that we should go forward and move on and do the NGS. So we concluded that we had a better test, higher dynamic range, gave us better sensitivity. And then in 2014, we moved our service, December 2014, to do all our translocation carriers by NGS. And since then, we've been doing reciprocal, Robertsonian, inversion carriers, complex translocation carriers. And another one which is really interesting is, because this is in our service and we validated it, sometimes we see people come through who haven't had a karyotype, who have no phenotype, and we identify consistent breakpoints in their cases, and they go back to their cytogenetics lab, and they are identified as carriers that didn't know they had their translocations. And we find a couple a month of those. In one instance, we found a complex recurrent pattern in a case, and we thought, oh, this is a complex carrier. And we went back, and the couple were karyotyped, and they were both reciprocal translocation carriers of 2 different translocations. So it's a very nice validation of the technology in a sort of blinded way. One of the things that is fun for me is you get these cytogenetics reports, and sometimes cytogenetics is written in an interesting alternative language. So you see things like this. This would be a report that you get through, a 4-8 question mark. Yeah, what the hell is that? That's the cytogeneticist going, well, I'm not really sure. And generally, you might then look at the the karyotype and go, Jesus, that's awful, and it looks horrible. And then what we do is we go back to them and go, do a better karyotype and come back and see us. So we get to do that. And what we do is, once we have a karyotype, we have a little tool that was developed 5 or 6 years ago by Bluenome that we still assess our breakpoints. And in this instance, we'd put it in, and it'd say a 518, and it would tell us the probability of detecting each of these imbalances, because it looks to see where these segments are along the chromosome, and it looks to see the minimum amount of DNA that we expect to see. So in this instance, we would expect to see for the 18, the end of the band and the length of the chromosome. So the end of the band is 71 megabases, and the chromosome is 76, so about 5 megabases. So we think there's about a 2/3 chance of seeing that imbalance, but the other imbalance is larger, so we have a high probability of detection. And you can go through these, and if we had a more accurate, so this has gone from 18Q22 to 18Q22.2, so it's like a higher level of cytogenetics, and we're able to use that to more accurately detect whether we're going to be able to see before we run the case. So this is before the case. And if they have the very end of the chromosome, then we can't determine without other information whether we're going to be able to see that imbalance, because it could be really small. So we might have to do further studies. And if you have both imbalances right at the ends of the chromosomes, then we struggle. And then we'd have to turn down the case unless we can get other information. And that happens in about, I think, around 1% of our cases. I haven't actually shown you the nicest picture. So what I did before this talk, I went through my mail, and I get sent cases to review. And this is what our last case, a care fertility case. And I just pulled it up and looked at it. And this was one of the first ones that came through. This is a 611 translocation. You can see the software's got better, so you very clearly can see those 2 imbalances. And in this case, you can also see at the end a trisomy 22. It shows that you can get imbalances and have a very low noise profile in the rest of it. Not wishing to join in too much in the joyful subject of mosaicism, but you do also see mosaics. Translocation carriers are not immune to it. They are just embryos. And in this one, you can see a chromosome 10 and probably a chromosome 17 mosaic. So we see that. And then, of course, you see cases where that embryo is either going to be balanced or have no translocation, but has other aneuploidies. So These are ones that with previous fish technology you might have implanted, but still clearly these have clear two monosomies here of 15 and 17. And joyfully, we do get embryos that are suitable for transfer. So this couple, I think when I looked through, they had 7 or 8 embryos, and this was their one suitable for transfer. And if we can find that, obviously they have a very good success rate. So when we get these cases where we have very small imbalances at the end of the chromosomes, what do? Well, we ask if there's any more cytogenetics. We ask if there's any products of conception, and we go back and use a microarray to try and refine those breakpoints. There's also the very high-risk cases. So these are the ones that come through and they have an affected child. And these are the ones you really have to be careful for from the PGD, because they're the ones that generally have the smallest imbalances. But we can go and take DNA from that affected child and we can run it to see how it works. We only do day 5 as well. The reason is people ask us and I say, well, you're going to get a better test with the day 5 than a day 3 from a technical point of view, so we just do day 5. I'll go through this a little bit quickly just to kind of give you the skinny, but from April 2014 to 2015, we have done 300 families, translocation carriers at Genesis Genetics. Average age coming through in this instance was 33. The sort of important numbers, the number of embryos that we call total euploids, so these suitable for transfer, was about 26% for all translocation carriers. So that's roughly where you are, 1 in 4 embryos suitable for transfer. When you break that down to inversions completely other subject that I'd love to bore anybody with if they want to talk to me about, and you get a better chance of finding something for transfer. In this case, just a few families, but just under half embryos suitable for transfer. If you look at the Robertsonians, so these are the ones where you get those little P-arms fused together, about a third, just under a third of embryos suitable for transfer. And finally, if you go to the reciprocal translocation carriers, this was our biggest group, 204 families, and a fifth of embryos suitable for transfer. On average, we have 6 embryos per cycle, so roughly just over 1 embryo suitable for transfer on average per cycle, and that's the expectation. There's no difference in female and male carriers, so I'll move on from that. The only difference we have is when we look at day 3 and day 5, it's more to do with the technology than anything else, and that's because we end up with more euploids. Well, they're day 5, they have less aneuploidy, but also we get more results from the day 5 than the day 3. So we have like a 2% failure rate on the day 5s and an 8% on the day 3. So you get a better result, more euploids, and you end up with a better number of embryos for transfer. And this is the sort of last slide about this, and this really just looks at the age of these age groups from 23 to 40, and the number of embryos that are euploid, as you can see, which is the second column, reduces from, this is our overall number, from around about a third. Interestingly, you always see it in genetics, and nobody really has a good explanation why the really young ones do worse in their 20s than somebody in their 30s. And you go down to about a fifth of them suitable for transfer. So just to sum up where we are in 2016, we've moved on because we have a better test, and that actually gives us more embryos suitable for transfer. It's a much easier workflow. I would love to stand up and give you our follow-up data. We're still collecting it. We hope to have it ready for publication later this year to really tell you about the miscarriage rate and how it's working. And finally, there's a picture of the Genesis Genetics team. A lot of the work has been done by Elizabeth Cameron, our genetic counsellor, who is in the middle of that picture. I can't show you where she is. And she is the one that's been collecting this data with her genetic counselling team. And they're the ones that we have, for me, the fascinating conversations about cytogenetics every week. So thank you for your time.